Hello all. Today we are going to start with a new chapter, that is Protozoa. So first I'll be talking about little bit about the morphology of Protozoa. All Protozoa are eukaryotic protists. So I repeat, they belong to the category of eukaryotes and they are found in the kingdom Protista, hence they are called as eukaryotic protists. They reproduce both by sexual and asexual reproduction. They are single-celled. They have an ability to move at least at some stage of their life cycle. They do not have a cell wall. They are microscopic in size. So the size ranges from 5 to 250 microns in diameter. Most of the protozoas, they are found in moist habitat. So they are either present in sea, soil or fresh water. Now most of the protozoa, they are found in a dormant state. Now this dormant state is called as a cyst. This dormant state is usually present when these protozoas are present in a dry condition, and this the protozoas do because they want to preserve their genetic material. So I repeat, protozoas are usually present in a dormant state called as a cyst. Now parasitic protozoa, they modify their morphology and physiology to cope up with changes in host. So the protozoas which are parasitic in nature, that means they require some kind of host to live. In that cases, as the host changes, the parasitic protozoa also changes their morphology to cope up with the changes in the host. Example of one such protozoa is the Malarial Parasite. Okay. Most of your protozoas are phagotrophs. That means they feed on particulate matter. So those are called as your phagotrophs. And protozoas are free living forms. Okay. So these are some points that you can jot down for your morphology of protozoa. Most of the protozoas have well developed morphology. They have a well developed cytoplasm, nucleus, plasma lemma, feeding structures, cyst and other protective structures, and certain locomotor organs which helps them in their motility. Now first we'll focus on the cytoplasm. Now cytoplasm is a homogeneous substance which consists of globular protein molecules linked together to form three-dimensional molecular framework. Inside this cytoplasm, you have some submicroscopic protein fibrils which are called as your fibrillar bundles. Now the bundles which are parallel fibers are called as myonemas or microtubule, and they are responsible for the contractility of your protozoa. In some form of protozoas, pigments are present inside the cytoplasm. These pigments can be in green, brown, purple or even rose color. Okay. Cytoplasm is differentiated into two types, ectoplasm and endoplasm. Ectoplasm is the outermost layer which is a gel-like substance, and endoplasm is the inside layer, which is actually voluminous and fluidy in nature. All your structures that are present inside your endoplasm. Now, what kind of structures are present inside the endoplasm? So, they are endoplasmic reticulum, kinetosomes, ribosomes, food vacuoles, Golgi complexes, contractile vacuoles, mitochondria, and your nucleus. Now, let's focus on the nucleus, which is present inside the cytoplasm. Now, all the protozoas contain at least one nucleus. Many protozoas, such as your ciliates, have multiple nuclei. Either the nuclei can be similar in nature or they can have dissimilar nuclei. In ciliates, two different nuclei, if they are present, one of the nuclei is called as the macronuclei, the one which is biggest nuclei is called as your macronuclei, and one which is the smallest nuclei will be called as your the one which is biggest will be called as your macronucleus, and the one which is smallest will be called as your micronucleus. Macronucleus controls your metabolic and regeneration process, and micronucleus controls the reproductive activity. Okay. Now, what are the structural elements present inside the nucleus? So, let's revise again. We have studied what is cytoplasm. Cytoplasm consists of two types, ectoplasm and endoplasm. We are now focusing on endoplasm. Inside your endoplasm is your nuclei. Now we are focusing what is inside the nuclei. So inside the nuclei you have chromosome, nuclear membrane and nucleoplasm. Okay. Next structure that we are going to study is plasma lemma. Plasma lemma. Okay. Cytoplasm covered by membrane is called as plasma lemma. So when your cytoplasm is covered by something from the outside, it will be called as your plasma lemma. The function of plasma lemma is to provide protection. It's a semi-permeable membrane and hence it controls exchange of substances. In some protozoa, plasma lemma is modified and have additional covering which is again used for protection, support, movement, etc. This additional layer is called as a pellicle. 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 Okay. In addition to pellicle, there are also some additional protective layers. So they can be thecae, that is THECAE. It can be in the form of shells. It can be in the form of loricae, that is LORICAE. Now this is made up of organic and inorganic matrix such as calcium carbonate and silica. Okay, so that was all about your plasma lemma and other such coverings that are present on your protozoa. Next is your feeding structures. Okay, now there are different feeding structures with the help of which protozoa can engulf food. These are sometimes called as dopodia. DOPODIA. Dopodia, which is present in amoeba. It is used for a pseudopodial kind of engulfment. The same like we have macrophageal engulfment, Dopodia does the same thing. Then they have something called as tentacular feeding tubes. So they have some tube-like structures that are coming out of the body. They are called as your tentacles, and with the help of that they can take their food. So this mechanism is basically present in suctorians. 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 Okay. In ciliates, they have a proper well-developed mouth. So this mouth is in the form of an oral groove. Okay. So oral groove, which is called as a cytostome, can directly take up your food. Or they have something called as food vacuole, which is called as your cytopharynx. Even in food vacuole, you can store your food and then later on engulf it. Now, they also have certain protective structures. For example, they secrete certain kind of mucilage from your mucocyst. Okay, they have certain tube-like structures from which they can release toxins. They are called as toxicyst. Okay, then there are something called as haptocyst, which when the haptocyst touches certain prey, that prey becomes immobilized. So it becomes easier for the protozoa to eat that particular food. So that particular process is done by a structure called as haptocyst. Okay. Now when we talk about locomotor organs, they have three basic locomotor organs. So they are pseudopodia, flagella and cilia. Okay. So what is pseudopodia? It is a temporary projection of part of the cytoplasm which do not have a rigid pellicle. So just like your microphage, it tries to engulf it. Flagella and cilia, they are fine filaments which extends from the cell. They can be 1 in number, they can even go up to 8. And cilia are nothing but short thread-like extensions which are coming from the cell, and it helps in the movement of the protozoa. Okay.